Thanks again for this invitation to come to this hot part of Earth from Toronto after the long winter. What I try to do today is just kind of summarize you technical aspects, a little biology, and apologize for those who are expecting to hear biology that we do with embryonic stem cells. But I think it was more important for you to see or present why we are still enthusiastic about embryonic stem cells, and how we see them as still something which still have potential to have further impact on how we do genetics in the mouse. And I don't really have to emphasize to you that in the last 25 years since embryonic stem cells, mouse embryonic stem cells are around, it just changed the genetic landscape in the mouse. So we do genetics now differently how we did 25 years ago. How I also see that there is some more additional things into the system, and I hope that at the end of this hour you will be convinced that we can still take advantage of this beautiful cell line that we have and do genetics even more efficiently, how we do it now. So let me just introduce you the embryonic stem cells. So where are they coming from, and what is this major property that we are playing with or taking advantage of it? So embryonic stem cells are coming from this embryonic state of the mouse blastocyst, 3.5 days after fertilization, the first differentiated structure. So this is where we have 3 different cell types: the primitive ectoderm, here these 10-12 cells, the primitive endoderm, these cells here, and the trophectoderm with the outside layer of this ball, and this is the source. And if you, as I said, this is the first differentiated structure. So if you project the cell types to the later embryo, you see where the commitments are. The primitive ectoderm cells, the yellow cells, are all end up in the embryo proper. The primitive endoderm cells are extraembryonic, forming the yolk sac, outside here, the yolk sac, for example. And the blue cells, the trophectoderm cells, all end up in the placenta, forming a major component, the trophoblast cells in the placenta. ES cells are coming from this guy here. So it's a permanent cell line, which is now established from this primitive ectoderm. And what I really just emphasizing whenever I can, that this is a completely cultural artifact, tissue cultural artifact. So there's no such a thing existing in the embryo during embryonic development, that embryonic stem cells. The origin here, the primitive ectoderm cells, are only there for less than 24 hours, and they are all gone. So ES cells, we have ES cells because we found the conditions, in vitro culture conditions, where these cells are frozen in time. They just simply divide and do not change. And that's what we are taking advantage of it in genetics. So one more important thing about these cells, that these cells retain the property of the origin. So they can differentiate into anything what the yellow cells can do if they are placed into proper differentiating environment, like back into an embryo. But they also have the limitation of the origin, so they cannot differentiate into the trophoblast cells of the placenta. They cannot differentiate into this primitive endoderm-derived cells, the yolk sac endoderm. So they retain the property of both the potential and the limitation of the origin. How we can test the potential? It's simple. We all know that when they are injected back into a host embryo, they can contribute into the resulting embryo and form chimeras. This is a chimera where ES cells are leg Z expressors, so we can stain the ES cell-derived cells in the embryo, and this is where they are, you see. And if you let this embryo develop to term and develop into adult, this is the typical chimera that you like to see when the ES cell contributes into the coat color, but this is practically the contribution in all the organs, the tissues in the resulting animal. And practically this property that the ES cells can differentiate everything into everything which is in the embryo or in the animal led to a technology. The technology is very simple, and then which is a genetic technology. So we take embryonic stem cells as a whole plate of 10 centimeter tissue culture plate. You have 10 million cells here, and you introduce the DNA into these cells, hoping that it creates a desired genetic alteration in some of the cells, but because they are large in number, so that you could actually, with a smart selection system, identify those which went through the desired genetic alterations, clone these single cells into a subline, take an embryo, and this is another way of introducing ES cells back into embryonic environment, when you take an 8-cell stage embryo and aggregate with a clump of ES cells, overnight culture to develop into a blastocyst stage. Now it's a chimeric blastocyst. It has ES cell component and host embryo component. And this embryo goes back into a pseudo-pregnant female, which gives birth chimeras. If this chimeras is a fertile chimera and have a component among the sperm from the ES cells, then mating this one with a female among the offspring, you see animals which are fathered by the sperm which is from the ES cell component of this chimera. So practically the DNA or the genetic alteration created here now end up in the mouse. So this is what we call germline transmission, so that when we have ES cells genetically altered, and then through this path we can get them back, this mutation, get them back into the mouse. So this is really a simple path or protocol that we do with all the genetic alterations that we are creating in ES cells. This is how we go back into the mouse. So the secret here, why it just becomes so successful in genetics, embryonic stem cells, there are two big issues here. We are dealing with large numbers. So it is 10 million cells in a 10 centimeter dish. That's the population of New York City. So certainly you have weirdos there, right? So, and you are looking for practically those weirdos where genetic alterations happened of your desire, like homologous recombinations based knockouts and so on. So the other property is germline competence. And these are the two things, large number and germline competence, which practically made this cell line that successful that you observed in the last, let's say, 18 years now since the knockout is in business. So this is what I said, so that gene targeting practically changed the landscape now, because we can do reverse genetics. So you have a favorite gene of interest, you have no clue what it does, and a very, very powerful way of finding out what's the function of the gene is you just simply knock it out and to remove from the genome. And as we know that this is it's a very, very simple principle based on homologous recombination. So your gene of interest, and then you have a target vector, which is a very, very special structure. There's 5' homology, 3' homology, and a selectable marker replacing an essential part of your gene of interest. And when this target vector goes in by homologous recombination, you can create a null mutation in your gene. So if I think, maybe it's not the correct number now, but this is the bulk line here. It's about 4,000 genes are knocked out maybe in the last 17 years. And I just want to give a visual support what I'm saying now, that it had an impact on our understanding of gene function and gene diseases. So practically, these are just taken out from publications where reporting gene knockouts. So it's enormous information what this 4,000 gene knockouts created now in our understanding. It was a major impact in our understanding of development and diseases. There are several ways of creating mutations, not just the knockout. So this is another very efficient, high throughput, I think, approach to create mutations in the genes, which is called gene trap, which is based on another principle, practically a simple vector, which has a so-called splice acceptor and a reporter and a selectable marker, fusion protein, the beta-GO. It's a LEGZ neofusion protein. And if such a construct lands somewhere in an intron into a gene randomly, that it could steal the expression of the so-called trap gene, and then you can get neoresistancy and LEGZ expression from that ES cell lines. And at the same time when it renders the cells to survive neomycin, it also mutates the gene when the whole thing is landed. So it's not that much of a kind of reverse genetic approach. It's a kind of random insertion, but now it's developing into a throughput level where you can get efficiently mutations from dozens of genes that we have expressed in embryonic stem cells. So homologous recombination-based gene targeting and gene trap-based random mutagenesis. Now, these two things came to the point where, as Ken pointed out, that now is an international effort which practically aiming in the next 5 years to target all the mouse genes and create embryonic stem cell lines where we have all the genes what we know mutated in different cell lines. So practically this effort is now a kind of combination of a European, a Canadian, and an American effort. UCOMP is the European, the NORCOMP is the Canadian, and COMP is the US effort supported by NIH. And this is pretty much the level where what we had when the issue was to sequence the human genome between 1995 and 2000. So it's a good amount of money placed in all the three different places into it, and the aim is to saturate with mutations the mouse genome either by gene trapping. So for example, the NORCOM is planning to create 144,000 ES cell lines with different gene trap insertions and 2,000 targeted ES cell lines. The UCOMP, the European, 120,000 gene trap and 8,000 targeted lines, and the American effort, 8,500 targeted cell lines. So if you just add together the targeted cell lines here, it is above 18,000 genes, which is at the moment the high quality annotated gene number. So practically, if there will be no overlap, but probably there will be overlap between the three efforts. The targeting itself could create a saturated situation where we have all the known genes are knocked out in the next 5 years in the mouse. So we will have ES cell lines, embryonic stem cell lines of thousands. And now the big challenge is after that, after 5 years, what we are going to do with this. So how we are going to phenotype all these mutations. And this is where we really have to think in the future so that it is going to be done by these three major efforts. So how we are going to get phenotype information in a way that is still feasible to do with our money that we are shrinking money that we have for research. With that, so that I would like to go to some of the details that we think that maybe it helps to accelerate things. But before that, I just would like to introduce you another tool, which is now built in practically all the three systems, is the recombinase. So I mentioned to you the homologous recombinase targeting, the randomization-based gene trapping, and that's an additional tool which is really very powerful and then give lots of mileage, I think, or ways to analyze phenotypes efficiently. And as you know, recombinase is it's an enzyme which acts on DNA and recombines between two attachment sites called LOXP sites. So the end of the recombination, the result of the recombination, which created by the recombinase between two consensus sequence LOXP site, is this another two LOXP site where recombination occurs here at the midpoint somewhere. This is the result. This is also a LOXP site. And then because the enzyme is quite efficient of recognizing the LOXP sites and quite specific also, so we are using this in both in gene targeting and gene trapping approach by doing these things here, that if you fortunately don't see this, there is a loop here, so the projector doesn't have enough contrast here. So, but the bottom line is that if you could see this very light circle here, that the recombination between two LOXP sites placed in the same DNA strand is the removal of the intervening sequence between two LOXP sites. So it's excision. But maybe with this one you can see what I'm talking about. So combining the homologous recombination with site-specific recombination allows us not just to create null mutations, which is very important, I say that, but also very important to have tools which allows us to create subtle changes in the mouse genome. And this is, for example, how we create specific point mutations in a gene. So by targeting this gene with a special target vector, which has a selective marker, as I showed in the middle, which are LOXP site flanked. These little triangles are LOXP sites. And then introduce the point mutation, desired point mutation in one of the arms, like the 3' arm itself here. And then when this target vector recombines in, delivers the point mutation into this exon, we are able to remove that disturbing neoselective marker, which is absolutely needed to identify the cell line, by simply expressing the recombinase in the ES cell line and create a so-called point mutant allele, which are absolutely normal exons except this point mutation, which is introduced into the target vector, and a little LOXP site left behind in an intron where the neo insertion was. So that is a very, very powerful thing. Other very powerful thing in ES cell-based mutagenesis is that we are in a position using these tools to create conditional targeted alleles, which when you have a gene of interest like this, now you can create another version of it, which is completely functional, by targeting this gene with such a target vector that you see here, where the neoselective marker is placed in an intron flanked by two attachment sites for another recombinase, which is called flip recombinase. And the LOXP sites are used to flank an essential exon of the gene. So when such a target vector goes in by homologous recombination, it creates an intermediate allele where the neo is sitting in an intron FRT site flanked, and an essential exon is LOXP flanked. And with the flip recombinase, which is just like recombinase removes things flanked by the FRT sites, now you can generate this allele here, which has an FRT site in intron, very short sequence, not disturbing, and the LOXP site flanking an essential exon. So such an allele of the gene of interest is still functional regardless of this little disturbance in an intron. But you also see that this functionality really depends on whether there is a recombinase around in the cells or not. And if recombinase is also expressed in the cell where such an allele is existing, then the recombinase itself can remove this essential exon from your gene of interest and create a mutation of your gene of interest, but only in the cells where the recombinase is expressed. And this is where the recombinase transgenic lines are coming into the business. So just wanted to show you some what we had in the lab. There's the nestin promoter driving recombinase expressing the recombinase specifically in the neuronal lineage in a developing nervous system. This is another recombinase where the recombinase is driven by the Ty1 promoter, deriving the recombinase in endothelial cells, so that recombinase now is expressed when not developing vessels and could create a knockout of your GNRS only endothelial cells specifically. This recombinase is expressed by nephrin-1, which drives the recombinase in podocytes in the kidney, and this one by the collagen 2 promoter, which drives recombinase in the chondrocytes and developing bones and cartilage. So these recombinases, as I see, that has this specificity. Now combining this with conditionally, you can also see the way how you can generate knockouts of your gene of interest, now cell type specifically. So if you want to knock out your gene in the brain only, you can use this recombinase and conditional alleles for your gene of interest, and then so on and so on. So the key databases, now it's an issue how to find this. I have one on my webpage, which is called CREXMICE. So we try to collect data on Cree transgenic lines made, so that if you Google up CREXMICE, you might be able to find, or you will find it because it comes to the top of the list. So there's nothing else called CREXMICE in Google, so just this one. So and then you can see It's about 117 Cree transgenic lines now listed there, and you can search the database for whatever specificity you are looking for. So combining homologous recombination with site-specific recombination now really changed a lot, because we are in a situation where I frequently say that now we are able to tailor the mouse genome. So not just mutating it, but whatever kind of change is identified, for example, in human disease, like in leukemia, specific translocations between chromosomes, specific genes involved in the translocation breakpoints, we are able to genocopy, I just made up this word for you, genocopy this into the mouse, right? Create a really as close as possible genetic model for that disease. Whether it is going to be a good phenotypic model or not, this is really the question of the similarity or difference between the mouse and human. But as far as the genetic model is concerned, this is really very, very close, because it could be exactly the same what it happened in human disease. So with this one, so that we are able to generate now subtle genetic changes, as I showed you example for point mutations. We can create knock-ins, we can replace genes for another genes, we can create large chromosomal aberrations, large deletions, reciprocal translocations, and we can do these conditional knockouts, tissue-specific and inducible knockouts, what I haven't mentioned, but I may be at the end show you some examples for inducibility things. So another important technology that we are putting in now, when we are thinking about using ES cells as a primary subject of genetics, is that we are using, as I said so far, that two properties of ES cells, the large number and the germline competence. The ES cells can do more. I'll show you that ES cells even can make a whole mouse, as it is. No other cells, but ES cell-derived cells. So let's just go back to this, what I showed, that if you take embryonic stem cells and aggregate with an embryo, and you can make these chimeras. That if you look at the ES cell contribution in these chimeras, you observe that ES cells can contribute quite well to the embryo proper, but their contribution, I mentioned in the very second slide, that it into the extraembryonic membranes is very, very limited, because they practically retain the limitation of the origin, so that extraembryonic development of ES cells is very, very poor. So we took this as an advantage and to try to benefit from that by creating this system here, where if you take ES cells and introduce into an embryonic environment when the embryo environment is compromised, compared to the ability of ES cells, just gently compromised. And that compromise was that we simply duplicated the number of chromosomes in mouse embryos, created tetraploid embryos as a host embryo for ES cell chimeras. And what happens in a situation like that is that ES cells, wherever they can differentiate into, they outcompete these lazy tetraploid cells, and end up with completely ES cell-derived lineage. But because ES cells can differentiate into all the lineages of the embryo proper, they outcompete the tetraploid host from the embryo proper. However, ES cells are not able to contribute into the placenta, so there's no competitor for the tetraploid cells there, so they develop into a nice tetraploid functional placenta. So with this trick, practically we are able to derive completely ES cell-derived embryos. And we showed this quite a while ago, 16 years ago, 17 years ago. And 3 years later, we also showed that some ES cell lines, those the R1 ES cell line we generated, could support the development of the embryo, but also beyond term, so and then end up with completely ES cell-derived animals. And you might want to appreciate it, because these mice are coming from the dish. So no father, no mother. So it's, however, the efficiency was quite low here, so that we got only 4 or 5% efficiency of generating mice from an early passage ES cell line. So that changed a bit after we turn into this new century. So it turned out from a study of Rudolf Jenisch's lab that ES cells derived from F1 hybrid embryos somehow has superior developmental potential compared to inbred-derived ES cells. And we generated a couple of ES cell lines from F1 hybrid embryos from between 129 inbred and C57 inbred strains. And we found, for example, some differences between developmental potential measured by this tetraploid complementation assay. And what you see here in this table is practically the result of this tetraploid complementation. For example, take this G40 ES cell lines. We made 68 little aggregates and transferred 68 different aggregates with tetraploid embryo into pseudopregnant females. And we got 30 newborns, completely ES cell derived. 26 reached adulthood. So 40% practically reached adulthood of the aggregates transferred. So we could make mice from these ES cells with this tetraploid aggregation with 40% efficiency. So, and this is which has really changed the way how we think about now embryonic stem cells. So you have a dish of area cells here, and as a half of an experiment, you have running mice from this dish, derived from this dish, now with this number. So obviously, this ability now changed how we look at the ES cells, and it's obvious now. So that why don't we just create mutant ES cells? At the loss of function, you can knock out 2 alleles of the same gene, or make RNAi, knock down your gene with RNAi, or create gain of function mutant ES cells. It's easy, just you have to express it, or whatever you want. And then take the mutant ES cells, aggregate with tetraploid embryo, and 2 weeks, and you have the things to analyze. So no waiting for germline transmission and F1 generation, F2 generation, genotyping, all 9 months, 1 year, just from the start, 2 weeks, and then you have things to look at it. And also, if you have your mutant ES cells, you can also take advantage of this developing field where we know more and more about how to differentiate ES cells into other type of cells, like into cardiac muscle, neuron, blood, skeletal muscle, bone, and then so on. So this in vitro assays also helps to speed up the phenotyping and analyzing and understanding the consequence of a mutation generated in ES cells. So to show this, that it works, that I would like to show you a pilot study or a proof of principle study just published in March in PNAS, where one of my students, Sophia George, actually took a kind of gain of function approach where she generated a sort of a conditional transgenic ES cells with this simple transgene promoter driving a LOXP site flanked stop region followed by a cDNA of your gene of interest. And as you see that because of the stop region, this promoter is separated by the cDNA. This transgene is not expressed as long as Cre recombinase is not around. But if Cre recombinase is introduced into the cells, the Cre recombinase removes the stop region, brings the cDNA under the transcriptional control of this promoter, and activate the transgene expression. So with this so-called conditional transgene idea, she generated a whole bunch of constructs with different cDNAs, and I show you the list of cDNAs that she was using in this conditional situation, and electroporated one each into ES cell lines and established a stack of conditional transgenic ES cells. And obviously these cells are happy because the cDNA is not expressed. And then she started expressing Cre lines, Cre transgenic, Cre protein in these cells by transiently expressing the Cre recombinase, and derived a daughter stack of this conditional stack where the stop region is removed and the cDNA is activated in the ES cells. We call it expression stack. And then when she has the expression stack taking each ES cells representing one of these cDNAs, then she took now ES cells aggregated with tetraploid embryo, and then she had the embryo to analyze 1 or 2 weeks after she set up that, she created the expression ES cells. So with this one, I just wanted to show you that it actually worked. It was a proof of principle experiment. For example, this was the embryos which she dissected where she expressed in a conditional manner, but these are now activated transgenes expressing the stem cell factor with a good old steal factor. And this is practically half of an experiment, and you could have enough embryos and females with these embryos that you can dissect a different type of the pregnancy. At 10.5 days, no problem with the embryos. 15.5 days, still the embryos are alive. 13.5 days after fertilization, the embryos still started showing some problems, hemorrhaging here and there, but they're still alive. 15.5 days, these are all dead. Embryos, they don't know further development. And so definitely enough to have enough material to look a little bit closer what's happening in these embryos. I just want to show you some, I don't know if there's a way to, maybe this one. Oh, not that. Any way to turn this on? This one? Oh, okay. Yeah. Speaker. Ammo. All right. Okay, good. So you see that at 13.5 days, so when we started seeing this hemorrhaging, the embryo looks really different than the Y type. And then when we looked, first I'll just show you because it definitely refers to something what I would like to actually, I'm going to talk about later, that this was a transgene that we use intentionally a promoter which is expected to be kind of overall ubiquitous type. This was the chicken beta-actin promoter with the CMV enhancer. Many of you know that. It's believed to be the best, strongest ubiquitous promoter. I show you where it is expressed in this particular transgene because this is the conditional version of this ES cells where not the stem cell factor is expressing, but still the LAGZ, because the LAGZ was the stop region. Remember that it has a stop region between the promoter and the steal factor, cDNA. So before the stop region is removed, it's expressing LAGZ because LAGZ was a stop. And we stain these embryos now generated with ES cell tetraploid embryo aggregation for LAGZ, where still the LAGZ was expressed. And we were quite disappointed because instead of getting a ubiquitous expression, you see a strong expression in the heart, in the somite, the brain area, but it's a kind of very, very sporadic in the genital ridge area, but it's certainly far from anything ubiquitous overall type of expression. And this is what you observe, so that the transgene expression, even you have this robust promoter, it's very, very much integration site dependent issue. And then you have to screen quite a lot of founders if you want to find the transgenic lines that you're interested in. If you do not have a control of the integration site, and then the transgene goes in randomly into the genome. But anyway, so this is what we got. But this expression actually, when the LAGZ is removed and the stem cell factor was activated, killed the embryo around 13.5 days, between 13 and 15.5 days, and resulted in this phenotype, which was quite interesting and amazing. So that in spite of the fact that it's lost the expression, this mouse is probably going to be a gold mine, because what you see here is that this is a Y-type section of the mouse, completely normal liver, completely normal heart, completely normal skin. And what you see here, for example, compare the skin of this one and that one, where the stem cell factor is expressed in this sporadic manner. So there is a layer here between the neural tube and the skin here. There is a clump of cells here attached to the heart. And then when you compare how the cells look in the liver, how the cells look around the developing vertebra or between the vertebra, and also the skin, just from the morphology, you see that, wow, it might be an ectopic hematopoiesis is going on here. So what is really exciting now with this mouse and then this is what we're checking now at the moment, looking for the hematopoietic activity of this cells which are not just came to these places which is not supposed to be, very much fit the liver-like cells. And then, in fact, this is what's happening, so that we have an ectopic hematopoiesis due to the fact of expressing the stem cell factor ectopically in this lousy manner. In the somites area, as I showed, it is expressed also in the skin, and also that heart area was also hematopoietic. So back to this proof of principle thing, so that this is the list of the cDNAs that we put into this second position and get activated. But we just to show that that assay actually it works, and we can get phenotype information out of the ES cells directly with this tetraploid complementation assay. We also had a couple of cDNAs where we expected no phenotype, like expressing human serum albumin or GFP or the DS-RAD in the second position. And all these actually showed that absolutely didn't get any lethality. So these activation of human serum albumin, GFP or DS-RAD end up with a normal mouse, not associated to any abnormality. So we are able to derive normal mouse from these transgenic lines. We were able to derive also normal mouse with ESL lines where the stop region is not removed from the transgene. So it's still silent. This conditional PAP, stem cell factor, VEGF-E, and conditional DS-RAD. Everywhere where the stop is removed and we expected by some biological activity, and you see lots of angiogenetic factor here, that VEGF-A, VEGF-E, FGF2, angiostatic factor, which is the soluble VEGF receptor, these two, IL-4, endostatin, and a couple of unknown. Whenever the cDNAs were activated, then we all found embryonic lethality. So it clearly shows that the group here, the neutrals, are okay to derive mice. Where the cDNA is activated, they all died embryonically at the different stages depending on the gene. So it was quite promising and quite convincing that the SSA works, and then you can get phenotype information out of ESL-derived animals or embryos. I just want to show you that, which was Christina made secretly in the lab, that this is one of the most beautiful mouse ever made in my laboratory, this red expressor lines, which this is how it looks from outside, but actually the organs are quite nice also compared to the Y type if you visualize the DS expression. So you see the testis, for example, the spleen, the kidney, heart, and so on. So, so with this, I would like to now switch gear and then show you another transgenic system that we really had high hopes, which is the tetracycline-inducible system. So we are putting the tetracycline-inducible system into ES cells because we want to use, again, ES cell-based genetics to understand gene function. And just to quickly go through the TET-on system, so how it works is that it's a binary system, binary transgenic system. It has a transcription factor called RTTA, driven by a promoter. And that transcription factor is tetracycline. It's an artificial transcription factor because it's made by the tetracycline repressor and fused with the VP16 viral transcription activator. So that transcription factor now has the property of binding DNA, recognizing the TET operator, and delivering the VP16 transactivator into that position where they find the operator. So the second transgene with the TET operator driving your gene of interest and transgene. Just because it's coming from the tetracycline-inducible system from the bacteria, the whole binding of that factor is tetracycline, or the derivative, the doxycycline-dependent issue. In the lack of tetracycline or doxycycline, this transcription factor, which is a mutant one, do not bind to the TET operator, does not drive the expression of this downstream gene. In the presence of doxycycline, that transactivator, transcription factor, binds to the TET operator and then drives the transgene downstream. So with this one, so the TET-on system, we knocked in to the RTTA, the transcription factor, into the ROSA 26 locus, which is a ubiquitous promoter, ubiquitous locus, the best ubiquitous locus ever found so far in the mouse, in a conditional manner, so that we have a ROSA locus here driving transcription, but it first hits a LOXP flanked stop region, which is the PGK neo selective marker. After this, again, the same idea how it was with the previous model. We place the RTTA linked with an IRES GFP just to see if the RTTA expressed from the GFP expression, which cells are positive for the RTTA expression. So this transgene is not expressing the RTTA as long as the LOXP flanked PGK neo is here. But if a Cre recombinase removes it, then the RTT is going to be expressed from the locus in the specificity of the Cre recombinase. So if it is a heart-specific Cre recombinase, then the ROSA locus expressing this RTTA only in the heart and nowhere else. So, and then you have the downstream gene here, the TET-O gene, driving your gene of interest. A little bit complicated because it's 3 transgenes, but you can see here very important things that we are taking advantage of the ubiquitous expression from the ROSA locus, but we are not expressing the RTTA directly, but that expression is Cre recombinase excision dependent. So Cre specificity will determine where the ROSA is going to express the RTTA. So we can take advantage of this Cre recombinase zoo, which is generated. Already hundreds of transgenic lines are made, right? And practically derive the RTTA with this specificity which is already created in the Cre zoo. So with this one, so that you also see that it's going to be then Cre specificity dependent plus doxycycline dependent expression of a downstream gene of interest. So this is what we now used as a base thing to develop a new system where we have now ES cell lines where we remove the stop region already from ES cell. So derive the daughter cell line from the original Cre line, or the original ES cell lines, having this ROSA driving RTTA, but in a conditional manner by removing the LOXP flanked neo. So in these ES cells, all these ES cells are expressing RTTA from the ROSA locus. Every cells are positive for the RTTA expression. Now we are electroporating into these ES cells a second transgene, which is this one here. The TET operator expressing something between the TET operator here connected with IRES beta GO, but just I remove this something here because I just wanted to see with the beta GO how this whole thing works. So with the beta GO, obviously that TET operator is a minimal promoter, doesn't derive the beta GO by itself if it lands somewhere. For expression of the beta GO, you need RTTA, which is there because the ROSA is expressing the RTTA, plus doxycycline. So we practically electroporate these things into it and add immediately doxycycline into the medium to make sure that that RTTA, which is generated by the ROSA locus, now it's going to drive this TET-O and gives us neoresistancy from the beta GO. So in the presence of doxycycline, we do a G418 selection and derive these clones, which are highly LegZ positive, surviving the neo selection. And then after picking quite a few of these surviving clones now, responding this way to the doxycycline. So if you add doxycycline to the culture medium, these double transgenic ES cell lines are all very nicely LegZ positive, strong LegZ for the surviving G418. If you withdraw the doxycycline from the medium, they turn off the LegZ in ES cells. So now you can imagine that if you have something, a transgene, inserted here, then practically you have a co-expression of the beta GO and your gene of interest in a doxycycline regulatable manner. So we have that. We show that it's not only working in ES cells, but also works in the embryo. So these are ES cell-derived embryos by this tetraploid complementation assay from these double transgenic ES cell lines. The difference between this one and this one that the mother got doxycycline in the drinking water at between E75, 7.5, and embryonic stage 9.5. The mother had this drinking water, doxycycline in drinking water, and then it turned on the LAGZ expression. And in fact, another gene here as well, because here we put the NMIG gene in front of the LAGZ. So these embryos are expressing not just LAGZ, but also the NMIG gene in all the cells. And if you do not have doxycycline in drinking water, there's no transgene expression in the embryo. Embryos are nice and fine. You see already the problem with the closing of the neurotube here due to the NMIG expression in the embryos. Just 2 days. NMIG expression in embryonic development. So it's going to be a very, very powerful way of analyzing the consequence of regulated inducible gene expression during embryonic development or in later stage in the adult mouse. So third thing, a lot of things, just time. So just wanted to show you another technology that could solve the problem of this, what we suffered with the random insertion, that to get a reliable transgene expression. And the whole issue goes back into the known fact that if you have no control of transgene insertion, you have an unreliable expression, and you have to screen a lot to identify those transgenic lines where you have the desired expression of the transgene. So and then it goes back to how to control the landing or the genomic insertion of a transgene in a 100% reliable manner in ES cells. And this is a so-called docking site and incoming vector idea. So practically the idea is that place down a site into the mouse genome, which we call docking site, which can be replaced in a manner so 100% efficiently with a replacer. It's a very, very clean way. So we can, for example, put a docking site into an existing locus that we trust, for example, into the ROSA 26 locus. And then after that, the docking site is designed in a way that if you go with an incoming vector and with a proper selection, you practically can replace in a very, very clean way the docking site to whatever you want to leave behind. This is the dream and this is the idea. And if you have such an idea, obviously you can actually think about that's really great, because now if you can target, for example, create targeted genes or targeted alleles instead of creating just a null allele, we put down the docking site into your gene of interest. And then after that, this docking site can be replaced to anything else. So you can put into this docking site to anything what you want to express by your gene of interest, like fluorescence proteins or recombinase or diphtheria toxin if you want to do ablation and so on. So lots of things that can opens up in a way immediately if you have such a system in place. So and how can you work out this system? So we try to concentrate on a third integrase, not recombinase, not flip recombinase, but a third one, because we want to keep the recombinase unused for later use. And this third recombinase or integrase was this one, the 531 integrase that we showed first a couple of years ago, that it works in ES cells. And the beauty of this integrase is that it recombines between two so-called heterotypic attachment sites. Heterotypic means that it's not the same sequence. So it recombines between an ATTP and an ATTB sites, and these are sequence differences between the two. The result of the recombination here is a hybrid sequence which are no longer substrate for the integrase. So that integrase recombines only once between two sites, attachment site, then it destroys the attachment site. So it's really created for pop-in type of integrations, because it does it only once and it stays there, whatever it popped in. So the idea here is if you have, for example, an attachment site placed down into the genome and a circular DNA which has the other attachment site, the result of the integration is that fusing in the circular DNA into that ATTP site which is placed in the genome. It definitely works, but obviously the efficiency is not very high, so that we have to make it very high by building in selectable system into this process. And the selectable system is quite simple, which really makes it 100% efficient, because the docking site is nothing but just the attachment site, the ATTP site, with a promoter-less selectable marker, like a neomycin resistance gene, no promoter. Such a docking site is just sitting there, it doesn't do anything, right? Because the neomycin, if it is a neomycin, doesn't have a promoter. So the incoming vector brings the promoter for that promoter-less selectable marker sitting in the genome. And if you see that this incoming vector doesn't have any selectable marker, just a promoter. So if it integrates somewhere else into the genome, it's not visible. The cells are going to die. So however, when it integrates, recombines between the two attachment sites, then it brings the promoter in front of the neo. And it drives the neo, makes the cells surviving. So that's in this system, the incoming vector, when there's no selective marker in the incoming vector, it's really 100% efficient, and you can generate, whoever survives the selection is this one. That happens, that if you go through this, practically you get this one. The promoterless neo is going to get a promoter, cell survives neomycin selection, and deliver the late replacer into the site. We have, after this, we identified this cell line on the basis of resistancy for the marker. We can remove it because we put FRT sites around this area. So, and then practically just leave the replacer behind. So it's a very, very clean replacement, 100% efficient as far as this is concerned. And now we are working also to the efficiency of this one, where we believe that it at the end is going to be 100% efficient from this to this, a complete clean replacement. So it's 100% efficient, as I said here. So efficiency of integration site-dependent docking site, this was an issue, so that we have this system now working. The next question was that, is the efficiency of going into a docking site the same for all the genomic sites, or there is differences? There are more permissive genomic sites for this integration and less permissive site. And in fact, this is the true thing. So this is what's happening. So there are different genomic sites have different permissiveness for that type of integration. So there are more permissive sites here, or this one, where you have lots of colonies after this integration, after the incoming vector insertions. And there are some sites where practically it's non-permissive for the 5C31 mediated integration. But for this purpose, obviously, we are not going to use this one. So the bottom line is that it works in most of the genomic sites that we looked, where we placed docking sites into. So if you just wanted to show you that this really works because it just went behind. So we replaced practically the docking site with a EGFP incoming vector, and then we got all the colonies that we have now expressing the GFP coming from the same docking site. We also have molecular markers where I could prove that they all went into the docking site, so there's no practically background issue here. And so far we checked 140 resistant colonies, and all were actually correct integration to docking sites. So that's just proving that this is really a 100% efficient way of putting a transgene into a predefined genomic site defined by that artificial piece of DNA which is placed there. And just to show you that ES cells are expressing not just the ES cells are expressing the GFP, but after that, when we make embryos out from this ES cells with tetraploid contamination assay, that all the cells which are derived from these ES cells are expressing the GFP, so that we have a technology where we can control the transgene expression and in a much better way. And if we defined the permissiveness of the docking site as far as the expression is concerned, we can get gene expression in a much more reliable manner that we could get earlier when we had to do random insertion of transgenes. We all need embryonic stem cells, obviously. We, at least so far, we were not able to do this with the classical transgenesis, when you inject DNA into the pronucleus, for example. So you need embryonic stem cells to have this. So, and the ability of generating mice now from ES cells, and those are these technologies now, which is now building around a new way of creating controlled transgenesis. We believe that's going to speed up tremendously the genetic research or genetic approaches in the mouse. Embryonic stem cells, making embryos and animals directly from ES cells, and all these new ways of controlling transgenic expression and insertion. So with all these tools, what the real challenge is that part of my lab is now switching to this ES cell line, which is human embryonic stem cells. And so we established 2 years ago the two first Canadian human ES cells, CA1 and CA2, and we are now busy to establish this transgenic technology, what we do out in the mouse and we have in the mouse now in human embryonic stem cells. Believing that genetics will be a great tool of making these cells behaving how we want them to behave, and making it safe and efficient basis of cell-based therapies in the future. So with this, I would like to acknowledge all these people so that my lab is very, very enthusiastic and keen on both developing technologies but also using the technologies for addressing biological questions. So Koichiro, Takeshi, Claudio, Gustav, Marina, Stefan, Ella, Jonathan, Sophia, Puzan, Christina, Paria, Hao, Luis, Magosha, Sue, Sandra, and collaborators Janet, Rosan, Davido, and Nishma Kasam. Thank you very much.
Thanks very much, Andras. I think we have time for 1 or 2 questions.
Going back to your slides, you showed the great data where the ES cells that were selected with G4 and 8 also have the beautiful lactate. I was wondering if that idea has ever been translated into checking for homologous recombination and for gene targeting construct in ES cells. If you could, instead of selecting with the drug, if you could use a reporter construct to check for a targeting event.
So you have a reporter construct where you,
instead of having a PGKE or you have in your construct, EGFP or a LAC-C or even maybe a beta geo, and use the fluorescence to select instead of a drug.
yeah, so there are efforts using also fluorescence proteins as markers. What I've seen so far is that putting the fluorescence proteins outside of the homology arms and using as a negative selectable marker instead of a positive. So like what you use for positive negative selection, TK outside. So that I've seen targetings where instead of TK, they were using fluorescence protein gene as a negative reporter. So then whenever you see the cells is green, you know that's not a homologous recombinant.
Do you know, is there more success with that kind of strategy than
I don't know. I know that a company was using it, but whether it was just a way of getting around the patent of positive negative selection.
The G4 is not necessarily obviously to cut down on the number of non-targeting or non-inserting clones. So your idea is to reduce the total number of cells that you actually end up plating.
Yeah, it also seemed like a cheaper and faster way to get to the, your 600 clones.
Should we be thinking about putting those docking sites in our clones that we're generating? It seems like a very versatile thing to have scattered throughout various genes.
So the NORCOM project is using the docking site. So practically our NORCOM target alleles are creating a null allele by replacing an essential exon with a docking site. So you can go back, for example, to this null allele and be delivering back the removed essential exon LOXP flanked. And then you can generate, for example, a conditional allele from the null allele by this way. But also you can put that list of things into it. So that's why we are quite enthusiastic about the NORCOM things, because I think it's going to be more versatile than the other alleles generated by Regeneron or the Sanger, which is mostly conditional, only a conditional allele.
Ours is actually sort of a gene trap knockout first, convertible to a conditional allele.
Right.
So it uses flip first to convert to a conditional, pre-conditional allele.
So our gene trap also using a docking site, so that is all both gene trap and the target alleles are replaceables.
What if you could have all three, best of all worlds?
Well, I mean, we're all doing this in a time frame that's overlapping, so we'll have you back in 3 years. Then we'll have data, you'll have data, we'll see what comes out.
Right.
Any other questions? Go ahead.
Yeah, I was wondering, is there a website where you could go in and find out if your favorite gene exists in a knockout form in a mouse ES cell line, and then you could just order that cell line to study?
So that's what you can see already. Actually, start seeing it, because the comp and also the UComp has a web-based search now, whether your gene of interest is in the list, where is it on the what position. You can also nominate your gene now into these centers. That yes, I would like my gene to be knocked out in the next 2 months, because that's and why. And if you are doing it now, so probably you have a good chance that your gene is going to be among the very, very first which are being knocked out now.
And then if you don't want the mouse but just the ES cells,
Yeah.
you can obtain those?
Yes, you can get the ESLs, obviously. So the mouse issue is going to be an issue still, so that because that's a different ball game to generate mice from these ESL lines. But it's going to be just like the gene trap, perhaps, that there will be places where you just either order the ESLs or the mouse. Yeah.
And actually, if you really just want somebody else to do it for you, I have a website that you can go to, www.gotmice.org.
Yeah.
And you have a little mouse with a cute little milk mustache on, and answer 3 questions, and you'll get a response back in 72 hours. 48 to 72 hours, if it exists somewhere, and if it does, I would guess it. Anywhere in the world. Any of the programs, past to present. www.gotmice.org. One more question? Okay.
Thank you. Good, thank you.